Okay, we are into roughly 7 days of fermentation for both our sauerkraut system and our cucumbers. And what we've been doing is enumerating with 2 days we did a plate count, and in 5 days we did a plate count. And what we're hoping to do is to follow the succession of the lactic acid bacteria as the system processes. And so the sauerkraut is 1 day ahead of the cucumbers, but typically the initial organism that we isolate in sauerkraut or cucumbers is an organism called Leuconostoc mesentoroides. It gives it a kickstart, produces so much acid, and then it will, if the acid gets high enough, it's going to die off, and the second group of organisms is going to pick up. Well, in this case, the Leuconostoc, we're going to show you a picture of it in a minute, is still around after 7 days, and we kind of thought about it. But so what happened, this time we're incubating it in an incubator and controlling the temperature. We incubated sauerkraut at 18 degrees centigrade. All the years before that we've done it, we did it at room temperature, which is 22 degrees. So what it means that this organism lasted if you slow down the temperature, you're increasing the time that organism it takes to make the acid to inhibit itself. So that kind of explains why the Leuconostoc is still around. Now, another thing, I have we have cucumbers over here, and we did the 7-day analyses on the enumeration. And remember that what we do is we take the sample, we do a serial dilution, we plate it out on growth media, and we enumerate the countable plate, which is the one between 30 and 300, or 250 and 200, and we come up with a number. So the other thing we did on cucumbers, we used 4 different selective media for enumeration. And what we did, we diluted the samples out at -5, -6, -7, incubated 2 days, and ironically what we found out that the countable plate is the -5, and the countable plate on sucrose was 132 colonies. The countable plate on tomato juice was -5, and it had 132. And the countable plate on the MRS had 132. So what does it tell you? We don't need to be using three types of media. We can cut that down to 1, and we're going to still get what we want. Now another thing I want to look at, I'm gonna work you backwards to find out what is the number. By counting this number, we have 132. It's at a -5 dilution. So the way we're going to convert that to find out what's in the fermentation chamber, we're going to invert the dilution so that it becomes a +5. So we got 132, and that's going to be 2 more zeros, and we're going to change that number to the scientific notation. So what we have at 7 days is 1.3 times 10 to the 7 per ml of each ml in that 20 liters of fermenting pickles. So you got to, we're gonna have you, don't know how we're gonna do this, but you're gonna, that is the cotton. And the other thing I want to point out, I'm stumbling around here, is that if you look, and I think you can see it on this camera, the type of colony, it looks like the same species. So we got, looks like 100% of whatever that is. We're going to take that, gram stain it, try to figure out, name it, so we can know what has succeeded the sucrose in that fermentation. So that's kind of how we're going to do it. So with that in mind, that's an update on the bacterial stuff. We're still reading the percent acid and pH on it, and which we'll make a chart with. So that's an update on the succession in pickles and sauerkraut.